In this lesson, we'll continue our discussion of the cellular level of organization by taking a look at the nucleus. So think of the nucleus as the largest of the organelles. We had already done the plasma membrane structure and function in previous lessons. We looked at all the different non-membranous and membranous organelles. This is kind of a membranous organelles, but I wanted to do it separate because it's so important. This is the largest organelle. Think of it as the brain of the cell, right? I like to think of it as the cell's control center. It's going to direct the production of all the proteins in the human body, right? So the nucleus is controlling when these proteins are made. The nucleus stores the blueprint for these proteins. It also determines the structure of the cell and the function of the cell, right? So without the nucleus, the cell wouldn't survive. All right, that's my next comment. A cell will not survive more than 3 to 4 months if the nucleus were to be damaged. Now, most cells have one nucleus, and if you looked at pictures or the microscope, right, we see the nucleus in the middle of the cell. But there are some cells that have multiple nuclei. Right, a skeletal muscle cell, known as a skeletal muscle fiber, could have hundreds of nuclei, and guess what, they're not even in the middle, they're more pushed to the sides. Where red blood cells, when they go into the bloodstream, they actually lose their nucleus. So when you look at them under the microscope, you don't see a nucleus. Yeah, so they're going to have a limited lifespan. So here's our picture from that composite cell picture. Right, here's the nucleus in the center of the cell. Again, we had done all these organelles in previous lessons, as well as the cell membrane. Let's take a look at the anatomy of the nucleus. So the membrane that's around the nucleus we call the nuclear envelope. Now this is not just one membrane, it's actually a double membrane, and it's actually attached to the rough endoplasmic reticulum at several locations. If you remember from this picture here, these membranes that are folded here with the ribosomes on it was the rough ER. That rough ER and the nuclear envelope in certain locations are attached to one another. They're connected. Now, between the two membranes, there's a little bit of a space known as the perinuclear space, right? So that's just the space between the two membranes of the nuclear envelope. This nuclear envelope is not continuous. It's not just one solid membrane. It actually has openings that we call nuclear pores. So these are communication passages in the nuclear envelope, and it allows for the passage of RNA and proteins from the nucleus into the cytoplasm. So let's take a look at these three structures. Here's the nucleus that's been removed from the cell. So you can see here, guys, this blue line, that kind of dark, and the blue line over here would be the double membrane, right? So that would be the nuclear envelope. Now, it's not labeled on this down over here, but it's labeled on the upper one. The space in between here is that perinuclear space. And then if you notice, there's these holes like right here. This hole is cut so we could look into it. But that hole right there, that hole right there, they almost look like craters on the moon. Those holes, here's the one that we have blown up, is the nuclear pore. Okay, so things that are made inside the nucleus can leave the nucleus through the nuclear pore and then go out into the cytoplasm. That's going to become important to us when we do protein synthesis. Okay, so the double membrane is known as the nuclear envelope. Within it are these holes known as nuclear pores, and the space between the membranes of the nuclear envelope we're going to call the perinuclear space. Okay, so we got everything on this slide: envelope, perinuclear space, nuclear pores. Here's a real micrograph, right? You can see it's magnified 9,000 times, and you can see all those nuclear pores. Look how many of them there are. Within the nucleus, we actually have a fluid. It's kind of like the cytoplasm that we see inside the cell. This fluid, instead of calling cytoplasm, you're going to call the nuclear plasm. And there are also, just like in the cytoplasm, support filaments. Remember this cytoskeleton? We have the same thing inside the nucleus to help to maintain the shape of the nucleus, known as the nuclear matrix, right? So this is kind of like the skeleton, the cytoskeleton of the nucleus. It has support filaments. Okay, so the fluid-filled portion you're going to call the nucleoplasm, and the support fibers inside the nucleoplasm are just going to be called the nuclear matrix. Now, if you look in the center of the nucleus, you may see one or a few, this dark area right here. This is known as the nucleoli. If there's multiple ones, we would say nucleoli. If there was just one, we would say the nucleolus. This is important. It's a little bit advanced right now. The main thing about this is that it's going to synthesize things that are related to protein production. I have that over here, right? The nuclei are basically related to protein production. And that's the main thing you should walk away with today. As we get into future lessons, we'll get into that in more detail. So the nucleoli synthesize or make ribosomal RNA. This is the RNA that makes up our ribosomes. So basically our ribosomes are born in here. The ribosomal RNA leaves the nuclear pore and then goes out to the nucleus, where the ribosomes are either in the cytoplasm or they're attached to the ER. Okay, so it makes ribosomal RNA and the different ribosomal subunits, the large and the small subunit. And as I mentioned before, they enter the cytoplasm through the nuclear pores. The nucleoli is actually made up of various things: RNA, some enzymes, and some histones. I'm going to talk about histones in a moment. These are proteins that are important in organizing DNA. But the main thing you should know today, as I mentioned, is that the nuclei, anything that they make, are related to protein production. Now, we tend to see more nuclei and more prominent nuclei in cells that tend to make large amounts of protein. Perfect examples of that would be the liver. Right? The liver is just constantly producing different types of proteins for us, muscle, even nervous tissue. Okay, so nucleoplasm, that's the fluid, again, inside the nucleus. The support fibers inside the nucleus are the matrix. The pit kind of looks like, it reminds me of a peach pit in the middle, or a cherry pit in the middle. This darker area here is the nucleolus or nucleoli, it depends if it's plural, and the nucleoli are related to protein production, right? They produce ribosomal RNA and the ribosomal subunits. And again, they are made up of RNA, different enzymes, and some proteins we're going to talk about in a little bit, known as histones. Okay, so these are all contents of the nucleus. All right, we have some more. Another content of the nucleus, most of us know this one. This is going to be DNA, right? We did this when we studied chemistry in previous lessons. DNA was deoxyribonucleic acid. And one of the main things we're going to see this semester in the next few lessons is that DNA stores the instruction, right? It has the blueprint for protein synthesis. It has the blueprint for every protein we make. You know, the pancreas makes insulin, right? The DNA has the blueprint on what goes into making the protein insulin. Now, our DNA, we had saw in previous lessons, was made up of building blocks known as nucleotides, and we had seen that there could be millions and millions of nucleotides. So our DNA is rather large structures. We need to keep them organized. So I'm going to go to a picture for a moment. If you look down here in the lower left-hand corner where my cursor is, I hope everybody recognizes this as a double-stranded DNA. All right. This DNA, we don't want it to become tangled up, right? We want it to actually stay organized. Otherwise, we're not going to be able to make proteins, or we're not going to be able to duplicate the DNA. So watch what happens. The DNA, if you follow it here where my cursor is going to wrap around these. There's actually 8 of these balls in here. These are proteins known as histones, right? We labeled them over here. And the DNA will wrap around the histones, right? So there's 8 histones, and the DNA will make these revolutions around it. This whole structure here is known as the nucleosome. So that helps to keep the DNA organized, right? So the nucleosome, and again, don't confuse that with nucleotide. The nucleotide is this, like right over here. This would be the building block right down here where my cursor is of the DNA, right? The nucleosome consists of these 8 histones and the DNA wrapped around it. All right, so the DNA coiled around the proteins. These proteins are known as histones, and they allow for large amounts of DNA to now fit into a very small place. So if you take a look now, these histones I'm sorry, the nucleosomes will form, and they will continue to kind of wrap around. And look what happens, guys. The DNA starts to become more coiled. Everything just becomes more and more coiled. When we see the DNA, if you look at this picture here, so that it kind of looks like messy hair. I always kind of say it kind of looks like straggly or messy hair. If we were to look under the microscope, we wouldn't even be able to see the DNA, which is kind of be like a dark area. This is the DNA, the way it looks in a non-dividing cell, a cell that is not going through mitosis, through cellular division. So if you were to look in the microscope, it would just kind of look like a dark area within inside the nucleus. And the DNA kind of looks like this if we magnify it. But when a cell is going to divide, it's going to go through mitosis. If you remember from previous lessons, mitosis is the division of the nucleus, right? Mitosis is the division of the nucleus. The DNA becomes even more tightly wound, super coiled. Watch this, we call this a super coil. The coiling just continues so much that we now get this characteristic chromosome that you've probably seen pictures of here. This is going to be the chromosome. This we can see under the microscope. Okay, so if you were to go under even 400 power or even 100 power, you're able to see the chromosomes pretty well. That's the same DNA as over here, down here, but this is what it looks like when a cell is dividing. So in a non-dividing cell, we call the DNA chromatin. In a dividing cell, the same DNA becomes more coiled, supercoiled, and visible. We then call it a chromosome, right? We call that a chromosome. But without all this coiling right here, all that DNA, which is this large structures, would never fit inside the nucleus. Okay, so let's go back to the notes. Again, the nucleosome is just the DNA wrapped around those proteins known as the histones. Right, there's usually 8 histones in those two that have the DNA coiled around them. Now the DNA, when it's in a non-dividing cell, takes on a more loosely coiled appearance. Again, if you were to look at the microscope, you wouldn't be able to see the DNA. We call that DNA then chromatin. When the cell is called to divide and the DNA has to duplicate and then split, we're then going to have chromosomes. Here the DNA, it's the same DNA, becomes more tightly coiled, more tightly coiled, and we can visualize it under the microscope. And again, we see that during mitosis. Right, so they do a good job with this. This is a good picture. Again, you could see again here the nucleus with the chromatin. It would just look like one kind of solid dark area, but here you would be able to actually see the chromosomes, right? And even in metaphase and in anaphase, early prophase also. Okay. This is just a blow up of the chromatin. I just blew it up. Right, again, you can see the DNA here coiling around the histones, forming a nucleosome, and then that becomes coiled, but not too tightly, and we call this chromatin, non-dividing cell. Opposed to in a chromosome, right? The chromatin now becomes more tightly wound, and we end up getting the chromosome. right? That would be in a dividing cell. So I just blew up the pictures a bit. Okay, so just to finish up on the nucleus, this is going to lead us nicely into our next lesson, which is going to be protein synthesis. Um, the nucleus actually stores information. All right, so this is about information storage in the nucleus. It stores information about proteins. The information that's stored there is made up of a sequence of amino acids. If you remember that proteins are made up of amino acids, and those amino acids need to be in a certain sequence. They just can't be random in order for that protein to work. That was the primary structure of a protein. So the nucleus contains the blueprint for the structure of a protein that is stored in the DNA. All right, and I'm going to teach you how that is retrieved. How do we get that blueprint when we go to the next lesson? Now, there's a certain language of the nucleus. It's actually the language of DNA. We call this the genetic code, right? This is the chemical language of the DNA instructions. This may be a little bit confusing now. Don't get too upset about this. We're going to do this. We're going to actually apply this when we do protein synthesis. If you remember from when we did chemistry, we have these nitrogenous bases that are found in DNA. Remember that the A stood for adenine, the T stood for thymine, the C stood for cytosine, and the G stands for guanine. So the genetic code is this, guys. A sequence of 3 nitrogenous bases. Let's just say A, T, and C. Forget the G for a moment. That sequence of 3 nitrogenous bases is known as a triplet, and each triplet will be the blueprint for one amino acid that's going to become part of a protein. So in DNA, when we see 3 nitrogenous bases, that will dictate one specific amino acid. Like I said, this is a little bit confusing. I'm going to apply this in the next lesson that we're going to do on protein synthesis. It will make more sense there. For today, for this lesson, just know that the triplet code is 3 nitrogenous bases, and these different 3 triplets, the different types of triplets, will determine different amino acids. Okay. And then finally, the last definition on this slide is a gene. The gene, for our purposes, guys, contains the instructions for the production of one protein. Watch this. Hopefully here on the right, you would recognize this as a DNA. Watch this. This box right here is one section of the DNA. If this section of DNA in here is the blueprint for one protein, this would be the gene. So let's just say this is the gene for insulin. This would have the instructions to make insulin. The next box down, let's say we drew a box over here, down here, and we made another box right here. This is another gene that may be the blueprint for another protein, right? So the genes contain the instructions for the production of one protein. In addition, they are the functional unit of heredity, excuse me. Our genes are different. We each have different genes. That's the reason if we were all in one class today and we were in the same room, we would look around, we would see, unless we were identical twins, we would all be different. So the gene also is responsible for what we look like, our makeup. Okay, so a gene is just one section of DNA, right? Here's one gene, right? Then this whole bottom half here could be another gene. They're going to be different sizes based on what they do. Now, if you watch this, they uncoiled the gene over here, guys, so we could see one strand on the right, one strand on the left. And then notice, remember that the base pairing A to T, C to G, right? A always base pairs with T, if you remember from chemistry, and C always base pairs with G. And then these dotted lines in between are hydrogen bonds. But notice here, this first sequence of 3, remember the CTC that we see here? That sequence of 3 is a triplet, and that triplet there is the blueprint for one amino acid. Let's just say, I don't know, let's just say valine. Then CTT, that triplet is the blueprint for another amino acid. Then AAT is the blueprint for another amino acid. So when we read DNA, we read everything as the triplet code, right? 3 nitrogenous bases will be the blueprint for one amino acid. Okay, and if we when we make a protein, we're going to see that this triplet, this amino acid that is from this blueprint, will be next to the amino acid that's part of this triplet over here. And again, I'll apply this for you in the next lesson. So just to give you an example, do not study this, do not memorize this, but just to kind of show you, here's a triplet. Forget all this stuff in the middle. Let's just look over here. Here's a triplet AAA, right? That means 3 adenines. Right, so somewhere along here, let's say we had 3 adenines. Here's one right here, AAA. That triplet right there, that is the triplet for a amino acid known as phenylalanine. AAT is the triplet code for leucine. ACA for cysteine. CCAA for valine. I don't want you to memorize these or anything like that. I just wanted to give you an idea that the triplet is determining what amino acid is going to be put into place, right? It's going to determine the sequence of the amino acids. Okay, so that concludes this lesson on the nucleus. In our next lesson, we're going to begin protein production. We'll take a look at a process known as transcription first, and then the next lesson after that will be translation.